And welcome to Draw My Science, the video podcast where I interview scientists and I make illustrations based on their amazing work. In this episode, I had a chat with Jennifer Honeycutt, Assistant Professor of Psychology and Neuroscience at Bowdoin College. Jennifer studies early life adversity in rats. She sees what happens when baby rats are separated from their litter mates, and how they can develop anxiety, or they can become brain-wired to recognize threat much earlier in their development than they should. Her findings have incredible implications, and they can help us understand what are the psychological consequences for humans when children are separated from the care of their family. We talked about the research and so much more. How she managed to work during the COVID shutdown, how she had to overcome challenges to become the amazing scientist she is today, and even how her research is shaping your own experience as a new parent. As usual, while you listen to the interview, you can see me drawing on screen the illustration I made for Jennifer, and you can find them in full resolution in the description of the video. Together with all the instruction you need if you want to participate to Draw My Science. But now it's time to talk about cute baby rats. Enjoy my chat with Professor Jennifer Honeycutt. Hi, welcome Jennifer to this episode of Draw My Science. I'll let you start with introducing yourself with your smartest speech.
All right. So for my research, I'm really interested in how early life experiences influence the trajectory of brain development and the development of mental health issues. So I do my research in a rat model of early life adversity via maternal separation. So we're modeling very, very severe caregiver deprivation, similar to what you would see in an understaffed orphanage. And we're looking at how that early experience, being separated from your caregiver and your litter mates, actually changes the connectivity between the limbic regions of your brain and the prefrontal cortex, and how that really can either make you more vulnerable to later risk and development of things like anxiety and schizophrenia, or maybe in some cases, maybe a little bit more resilient. So most of what we do is looking at males and females, trying to understand sex differences within the entire kind of realm of early adversity. Most of the outcomes are sex specific. So for instance, schizophrenia is more likely to occur in males versus anxiety more likely to occur in females. So what is it about this early life risk period that can kind of diverge, right? Brain development in different sexes to lead to different outcomes.
Okay, so you are looking at physiological difference, I assume. So really looking at brain scan and something that is happening because you mentioned some parts of the brain that are more separated. So is that in terms of connection between the two sides of the brain?
Yeah, so I look
and what do you use to measure that?
Yeah, so we use a couple of different things. So I primarily use neuroanatomical measures. So we do neuron track tracing, and we look at axonal tracks from regions of interest. So in this case, I'm usually looking at the amygdala and looking at how many of those axons and when do they arrive in the prefrontal cortex to tell a signal to the prefrontal cortex that there might be potential threat. And what I see, at least on the neuroanatomical side of things, is especially in females, if you've experienced early adversity, those connections arrive too early. So they're arriving and they look like adult patterns of innervation when they're juveniles. And we don't see that until like adolescence in males. So it's a little bit delayed in the males, which is pretty interesting. So you see that sex difference neuroanatomically. I also do a little bit of fMRI work. So we look at the rats that have experienced adversity in the fMRI, and we're able to look and see what the functional connectivity of those brain regions are, and if those are also in line with the trajectory that we're seeing for the neuroanatomy. Because the axons can get there, but we don't know if they're functionally integrated yet. So the scanning kind of helps us to see if they're functionally integrated.
And then my other very curious question, how do you know when a mouse is schizophrenic?
Yeah, so obviously,
or any other mental disease for what matters.
Right. So that's kind of the biggest trick in animal research, is that we can't really recapitulate an entire mental health disorder, right, in an animal. We can try and get a couple of symptoms, but for the most part, we look at symptoms, right? Mm-hmm. So what are symptoms and behaviors that we can infer have something to do with brain changes? So for schizophrenia, for instance, we can look at cognitive impairment. We can look at issues with prefrontal inhibition. We can look at issues with hippocampal processing. And in anxiety, we can look at anxiety-like behavior in an open field, or we can look at introducing something that could be potentially threatening and looking at like a hypervigilance response. And from that, we can kind of say, okay, well, they have this symptom subtype, and this is kind of what the brain correlate of that is. So it's very truncated. It's not a holistic picture, but it's as close as we can get, right? Because we can't just ask them how they're
feeling. Yeah, of course.
The hardest part.
It will be cute to have this mouse in psychotherapy, lying on a chair and talking with
right, yeah, and telling you something. And we unfortunately can't do that. We have to just look and guess and hope we're right, yeah.
No, but it's great that you have these markers, because I didn't know there was this possibility. So you correlate these more sciencey, traditional science approach of direct measurement, if we can call it like that, with the brain activity and everything, to a more psychological, sociological approach in which you actually have to observe the mouse and guess what's going on, what symptom they're having. Is that, did you need a different type of training for that, to get to that level of skills?
Yeah, so my background, so I have a bachelor's in psychology with a concentration in neuroscience, and my PhD is in psychology and behavioral neuroscience. So I have a pretty broad psychological training and background. I took a lot of clinical classes in graduate school, mostly because I'm really interested in clinical implications and translational models, being able to kind of better model these disorders in animals without missing big chunks, right, of what we know, and being able to actually compare what we find in a model of an animal to a model that we would see in a human, and trying to make the tests that we do as close as possible to kind of approximate what we would do in people. So I think my training and really kind of broadly in psychology, as well as the kind of neuroanatomical training that I got, was able to kind of allow me to ask the kind of questions that I ask.
Wow. And when you do this type of study, I guess there's a lot of variability. So what numbers are we talking about before you get to a point that you say, okay, I'm seeing this difference?
Yeah, it depends, right? So for the most part, whenever we go and we do an experiment, we kind of do power analyses and all that beforehand to be able to determine, all right, how many animals are we going to need to, if our hypothesis is correct, to see an effect without accidentally, right? We don't want to overinflate it, because if we were to just kind of keep going and analyze the data as we go and just keep adding more subjects, right? When we're like, we don't really see anything yet, add more subjects. Right, that wouldn't be an ethical way to kind of go about it, because then at that point we would just be fishing, right, for data. In order to kind of, usually at least the kind of research that we do, since we're using males and females, we have early life adversity or control, and we look at different developmental time points. We need a little bit of a larger comparison group. So we usually have like 10 animals per group on average that are typically, at least in our experience, sufficient to get a pretty good idea if a trend is sticking.
Okay. And then do you make adjustment on the group type to strengthen the result if you see a trend, or that's the final number, the one you decided to start?
Yeah, so the one that we decide on, unless there's like some sort of published reason, right, why we could go back in and edit it, that we could cite, We don't typically go back in and add things unless there's like a huge outlier and you go, we go back through our lab notebooks. Okay, what did something go wrong in the surgery? Was there something weird about this particular group of animals? And if so, then, we might need to substitute in one that didn't experience something that was out of the norm.
How did you come up with this idea for the study? I mean, did you look for other literature before, or it was something that you studied in psychology before? How did you decide to focus on this specific topic?
Yeah, so or at least the study I was talking about earlier with the connectivity in the fMRI. So that project I started when I came into my postdoc. And so that was almost 5 years ago now. And so we worked on that data. It was a big study. We just had it published in January.
Oh, so wow. Congrats, by the way.
It was a very long study with a lot of animals, a lot of surgeries to do the tracing and then all of the behavior and all of the fMRI. Coming in on that, a lot of that was part of my mentor's R01. So she kind of had a lot of it already kind of thought through. And then we adjusted some things kind of as we went and as we were like, okay, this is working, this isn't working. Maybe we need to adjust, the time frame for this. So we piloted it. Once we felt comfortable with placements for things and all of that for the surgeries, then we carried out the experiment. Much of that was kind of driven through my mentor, but I've kind of done an offshoot of that now. I have my own funding in the lab, and I do something similar where I'm still looking at cortical limbic circuitry and looking at amygdala reactivity as well as the prefrontal cortex. But I do it in awake animals in the fMRI, and I'm looking specifically at whether we have an increased likelihood of hypervigilant behavior if we give a potential threat. For this particular study, I was really inspired by a lot of work that's being done out of Nim Tottenham's group. She's down at Columbia. And they did a study a few years back that looked at kids that had been raised in orphanages and then adopted. And so they followed these kids out. And they did this thing called the fearful face task. I'm not sure if you're familiar with it.
No, go ahead then.
Yeah, so it's a really cool task that you can do with people. So you're in the MRI, and the experimenters will show you a picture of a fearful face, a happy face, a neutral face, and then they can look at what your brain is doing when you see these faces, even just if they're flashed. So it's a potential threat, right? We are really good at looking at faces and determining whether we should be worried. So I was really kind of inspired by that because they showed that kids with early life adversity show connectivity patterns when they see that fearful face that look like older kids, right? They don't have the typical kids, so they're almost wired. Their brain is wired to recognize that threat really quickly. So, and we see that in kids with anxiety disorders, this hypervigilance. We see it in adults that have anxiety disorders or PTSD. And so I was really interested in, okay, well, how do we do that in a rat? Because that's the model system that I use. I can't show rat faces to them in the magnet. So I had to get a little bit creative. And so in order to kind of do that same paradigm and then ask a similar question, but see, are we able to do what we see in people in the rats? Like, is it an accurate model? I built these ultrasonic headphones. So I have these tiny rat earbuds that go into the fMRI with the rats. And so they have them in their ears, and I can play back ultrasonic vocalizations to them. So rats make these ultrasonic sounds that we can't hear. One range is an alarm call that indicates potential predator, potential threat. And some are like giggles in a different range where it's like if you tickle them, they'll make these, or if they're playing. And so I play these back while they're awake in the magnet, and I look at what's different in terms of brain activity. So what areas of the brain are active if I play an alarm call versus giggles, right? Like what's different?
That was a clever solution.
So we've gotten the kind of the legwork down. We see different brain regions light up depending on which sounds we're playing that are kind of congruent with what we would expect to see. So like amygdala is really changed when you do the playback of the alarm calls. but you don't see any change in the amygdala when we play back the giggles. So we're really excited about that. And so now it's just implementing the early life adversity part of that, which I was supposed to be scanning. Actually, I had to cross it off of my calendar. I had a big cohort of animals I was going to have come in so I could scan them last week, which of course, can't do right now. I'm excited to see whether we'll see differences in those animals. And I think we will see that they're a little bit more reactive to them if they've experienced adversity, and that they'll show that a little bit earlier.
Talking about the fact that you're not in the lab right now, and you're working with corona from home because of COVID-19. Mm-hmm. I have a few questions about that. The first is, how are you managing the animal facility from home? And is there some type of advice that you want to share with similar researchers that are in a similar situation like you, that are working with that? Because, for example, I work only with instrumentation in my daily job, so it was fairly easy to put it on hold. Of course, it's not ideal, but the repercussions, they are limited compared to compared to someone working with animals, so that you've been planning this experiment for a long time. You want to be separating the litter from the mother at a certain time. So how are you working around that?
Yeah, so sadly we couldn't really work around it. So we had a couple of litters that were already like almost weaned. So they're almost like old enough to start doing work with. So we kept those litters, and my boss and I, my PI, we went in, and unfortunately our graduate students, we've got 2 graduate students and neither of them have cars. So they were and we're in Boston. So they were relying on buses and the T to get to the lab. And we didn't feel safe putting them in that position, right? So we had cars so we could get in if we really needed to, but we basically put everything on hold. Thankfully, the students had a lot of their data already collected. So they've been doing a lot of data analysis kind of at home and working on papers from that data that they did have. I'm in a kind of a weird position because I had just published that paper, so that was a big thing that I had been working on. I just started a new study, and I'm in the middle of another study, and so I had to stop the study we just started to look at kind of more of the neuroanatomical activity that was happening in the brain in the same circuit. So that's on hold until this is all done. And then I had to kind of cancel all of the MRI spots that I had because I'm unable to grow up the animals that I need right now to run them. So, and I don't have the manpower because I can't go and run it all by myself. So I'd have to bring in other people. So unfortunately, I'm kind of at a standstill collecting data, which is stressful, but I've been trying to use this time to like catch up on other things that maybe have fallen away, catching up on literature and things like that. And I'm also teaching a class this semester. So that's been keeping me pretty busy, transitioning that to online. But yeah, other than that, it's been a lot of kind of, more on the reading and writing side of things, working on grants. Yeah. Now I'm like, hey, I had time to work on a grant. Okay. So I can do that.
And for when you will be back, basically you'll need to apply for new animals, or they will.
Yeah, I don't know how it's gonna look. So it's gonna depend. So I actually I'll be finishing my postdoc at the end of July. And I got a faculty job that I'll be moving into. And so I was hoping to actually have all of this data to take with me so I could be analyzing and while my new lab gets set up, which sadly I will not have that now. So really at this point, it's trying to think of contingencies for in the fall, if things are back to normal or more so, how can I come back down to Boston to finish some of these projects because I'll be in Maine. So it's like 2.5 hours away. So it's not like too far. Like I could theoretically drive down and, do something for the day. So it's trying to figure that out. Yeah. And then also with the baby and everything that I got to figure out how maternity leave is going to look this summer. So it's a lot of things are up in the air right now, which I don't really have any answers for.
Yeah, no, I can totally understand. And but you said you're keeping busy, you're having your class. Is there any advice that you're like sharing from your experience working from home?
But I mean, I think the best thing is flexibility, right?
Mm-hmm.
Like, and I think that that's been, at least for my class, is kind of my go-to. Everybody's dealing with everything right now, right? It's especially, you know, and everybody's back everywhere across the world. So I know that I have to be flexible with deadlines, not only for my students, but also, you know, for me, because I'm juggling a lot of things right now.
Yeah.
And I think we all need to be a little bit more allowing of ourselves to be flexible. And to just, you know, sometimes we just don't have the time to do it because we're trying to figure out how to, you know, post this one thing or this one lecture and it's just not working for some reason. And, you know, it's going to take a little bit more time. And with the students too, just like being compassionate with them because they're stressed out too. They've got multiple classes. I'm teaching one class, right? I can't imagine taking like 3 or 4 classes right now, all online, all asynchronous, and trying to complete exams and all of that, when it's, you know, also dealing with, you know, being at home and, you know, not everybody's back in a great space. So it's just being cognizant of that, I think, is really important. But yeah, flexibility has been kind of my like go-to number one. Like, we just need to be flexible.
Yeah. So you mentioned before we started the interview and a few moments ago that you're expecting a baby with your wife. And how do you think your research is gonna affect the way you're gonna handling that situation is coming up?
Yeah,
do you think it's gonna have an effect at all, or
it's already had an effect.
Oh, really?
Yeah,
okay, tell me.
So funny. So we actually did reciprocal IVF. So my wife is carrying my egg, and we got a donor. And so one of the reasons that we did that is originally the plan was that I was going to carry her egg, but things didn't work out on the job market last year, or 2 years ago now, I guess. And so I was back on the job market this past fall. And I was very stressed about being back on the job market and did not want to have to carry a baby and be just really high levels of support all the time. I've done a lot of research in the past on kind of prenatal stress. And so I'm like, I've seen what prenatal stress can do. And like, I want to just like, we should avoid that if we can. We're already doing reciprocal, like my wife will carry because she had a really like chill semester. And so we decided to do that. And then of course now with everything happening, I'm like, well, we're all just stressed anyway. I guess it didn't matter. But at least now it's just like the last part of it.
The last part of the
whole beginning of it. But yeah, it'll definitely be interesting. I think doing developmental research and like I'm maybe too acutely aware of risk factors for different things. And so I know that we're always like constantly kind of thinking about that. We're very careful during when Christina was pregnant for, you know, gestational immune activation, because that's a big risk factor for later mental illness in some cases. So we were really careful about making sure that she had her flu shot and that we didn't go anywhere like that. Now with, we were actually very, very concerned because we were hearing some initial things where in some cases, if the mother was giving birth and she was positive for coronavirus, they were in some cases taking the baby from her for 2 weeks or limiting contact. And so that made us very worried. And given that I do caregiver deprivation work, that of course, like every red flag in my head was like, oh my God, like how, like we cannot get sick because
and that's that separation is important even in those so early days.
Even in like the first few hours, like it's so that I think made me the most panicked, because some people are like, oh well, it's good they won't get infected. I'm like, okay, well yeah, but like the psychosocial like bonding that happens there, that literally needs to last them their whole life as well. So it's like, where do you put the balance? Is hard because obviously you don't want the baby to get sick, but if you take some, and I was actually reading an article last night where it was a hospital in Brooklyn, there was a woman who had, and I think that she was still able to breastfeed, but she was wearing like a mask and washing her hands really well and everything before doing that, and I think that the outcome seemed okay for the baby, so that's good. But yeah, just kind of not knowing and knowing what can kind of be the long-term consequences of that was definitely been stressful for me.
I can only imagine.
Mm-hmm.
And going back to the thing you discovered or you found in your research, what's that thing you expected the least in doing your research? And that can be even in a sense of what you found very funny that you didn't expect, or very stressing that you didn't expect. It doesn't have to be necessarily a science result, just something about your work.
Yeah, I would say probably especially with that particular study, I think what was most surprising for me is a lot of times when we're doing, new methods. So I was learning fMRI for the first time, and doing that and trying to figure out, what are the ins and outs of getting an animal in an fMRI, in a restraint, and learning all of that. And, that I think was probably the biggest learning curve for me, was learning how to load them and get the fMRI working to get scans. And we did that initially, which I hadn't really thought of, but we did all resting state in this first study that I did, which basically means that they're like low level anesthetized for most of it, so they stay still.
Mm-hmm.
And so you don't really think about, when you're doing an MRI, like we can follow directions.
Yeah.
Still, but rats can't, and that's kind of scary, and it's loud. It's very loud in an MRI, which you don't really think about. And so having them anesthetized made it definitely a lot easier, but you can only get resting state connectivity from that. And so moving into my new kind of line of research where I'm doing awake imaging, now that brings a whole other thing where you have to habituate them, so that way they're not stressed out when you put them in and they're awake, because they still have to be restrained. So that way they're not moving around, you can get a good picture, but also we need to make sure that they're not like too stressed out. And they hate being restrained, which is understandable, right? They don't like it, but after a couple of days, they're like, oh, okay, yeah, like this is fine.
Nothing is happening to me.
Yeah, nothing's happening. I just chill out in this like box in this restrainer for like 20 minutes. Like that's fine. And we also play, we use like a mock scanner, so we play sounds, so it sounds like they're in the MRI.
Yeah.
To get them used to the sounds as well, and then we're able to put them in, and they're pretty calm, considering when we put them into the magnet. So I think that that learning curve was interesting, and really kind of just experiencing that was weird. Like kind of getting them ready. You don't realize. I think when you read some of these papers, like, oh, this is what they did, but like, how do they actually get that to work? Getting that to work has been like a whole thing, and building the headphones was a whole thing. Like that was pretty comical. That I worked with this one man at they do human audio stuff, so it's all human stuff. And so I contacted a bunch of people. Nobody wanted to make rat headphones at all. They were like, no, we're not going to do that. And I was like,
we didn't need
engineers for this. And I found one guy that wanted to do it, and so he was doing that, and he obviously didn't have access to the rats. And so he had gotten, I think, like a stuffed rat, like a fake animal, just to like show me where they would be positioned in like pictures via email. So I have all of these like pictures from my emails of like a stuffed rat with like these little like earbuds. Be like, is this about where you want them? That's where I want them.
That must have been an adventure. Okay, I'm gonna go with the last question about research, and then I'm gonna pick up. I mean, it's been a mixture in this interview, which was very nice. But I have a couple of questions more about your career, and but before going to that, I can easily understand the importance of the research. So I typically ask the people I interview why people should care, and it seems fairly obvious to me. So my question is going to be a little bit different at this point, is that, of course, give me your pitch of why people should care, but at this point you're basically following up on studies that have been done on children, on human babies and stuff like that, but trying to understand the why.
Mm-hmm.
So how do you think your contribution is going to change the result we have seen, and what do you think is going to be the next step?
Yeah, so I think So obviously the why should we care, right? Anytime there's growing up as a child, things are vastly different for different people around the world. And so environment is incredibly important, and caregiver interactions, whether it's a mom, dad, two moms, two dads, grandparents, whatever, right? That is critical for kind of accurate and timely brain development. And the lack of that, or any sort of addition of things like trauma to a child, is going to present a big risk for later on, especially during times where the brain is developing. And so for me, trying to understand what the underlying reason for that risk is, so what is the biology that's under that, is important to be able to inform treatment for humans, later on. So how can we do an intervention if we've identified somebody who's at risk? Is it possible, or do we need to be thinking about it even sooner? And by doing the research in rodents, we're able to kind of do systematic manipulations to pinpoint and try and find causality, as opposed to in the human work where a lot of it is almost purely correlational, right? Unless you have postmortem tissue. Really being able to pinpoint this is different here, and then pretty reliably this is different over on this other end, right? Then we know that there's some sort of relationship. And then kind of going from that, we really need to be able to have tests in our animals that can approximate what we do in humans, so we can actually compare them. So if we have kids that have experienced adversity and we want to do the fearful face task, right? How can we do that same thing, or as close as we can, asking the same kind of ethological question in a rat or a mouse, that is going to allow us to then be able to predict what we might see in a human scan later on. So I think that kind of the early work that I've been doing with the headphones and doing this kind of fearful face task analog, it will be helpful in kind of bridging this big gap that we have between what we're doing in the animals to understand human disorders and what's happening in human disorders. There's this big kind of area where, we have everything in rats here, everything in humans here, but like, can we compare them? And trying to find tasks and behavioral tasks, as well as fMRI tasks that are analogous, I think is the way we can do that.
That sounds fantastic. You mentioned you're at a turning point in your career. You're starting to have your own funding, and that's great. And you've had an interesting career from what you've told us, like starting from a bachelor in psychology and then getting more into the art science. So in this overview of things, what do you think has been one of the biggest challenge career-wise, and do you have any advice for young researchers that want to study something similar in your field?
I think for me, some of the biggest challenge, or really kind of the biggest challenge that I think is always kind of looming a little bit, is I'm a first generation college student. So I, from very early on, my parents were like, college is expensive. You don't need to go to college. We didn't go to college. Like it's fine if you don't go. But I was kind of hell bent on going to college. So going to college and kind of stumbling through, especially at the beginning with trying to figure out what I wanted to do. I started as an art major, switched to psychology and neuroscience, and then was kind of like, okay, well, what do I do next? And I really kind of struggled with what my next step would be after college. And I decided I would get my PhD because I was still really curious about the kind of neurobiological basis of things, and wanted to specifically do a PhD to learn more about kind of the brain anatomy and all of that. But not having anybody at all, even in my extended family, that's gotten a PhD or has had to deal with like student loan payments or applying for academic jobs or anything like that, it was kind of hard. I had to kind of build my own academic family that could kind of help me navigate, right, like the application process, and like, when should I be applying for funding? When should I be publishing all at once now, or should I space them out? Should I be, all of these things that are kind of, I don't know, like silent rules in academia that you don't really know unless like you're from that world. And not being from that world, I think I'm constantly kind of questioning myself even still, like, am I doing this right? Am I making the right choices? Am I applying to the right things? And I have to just kind of trust myself that I think I know what I'm doing, and being able to, the mentors that I've kind of found along the way. And I've been incredibly lucky to have met a lot of amazing mentors on Twitter. I've been on Twitter for a really long time. And since like 2009. So yeah, since I think I was an undergrad, I've been on Twitter. And so like world famous scientists are literally at your fingertips on Twitter. And it's been kind of wild to be able to be like, I have this idea, or I want to do this, and have those people support you or ask you if you need help with something, or, you can reach out to them and it doesn't feel as intimidating. I found that to be very helpful with dealing with the fact that I, need that extra support, and to try to find it, as Twitter's been like insanely amazing for that, which I guess, you would normally think of like a social media platform to be academic.
No, and I'm in a similar situation in the sense that I'm also first generation. How is that called?
Like first generation college.
Yeah, I mean, I come from Europe, so the idea of college is slightly different. We don't have big loans to pay, thankfully, but there's a lot of unknown because I don't have anyone in my family who has done it before. They have no idea what to suggest and recommend when you feel at loss. But I never thought about reaching out to Twitter for that. So that's actually a piece of advice that I could use myself. So thank you for sharing that.
I encourage students to do it. I'm always like, get on Twitter, like make a professional Twitter account, so that way you can, reach out to people and be like, this is what I'm interested in studying. People will find you.
And continuing that, and this will be my last question, your social media outlet and these type of things, how do you think are important, not only for reaching out to people, but also to communicating outside the bubble of academia what you're doing? And are we actually succeeding? Because I'm always concerned that the people following me, they will be other people that they're already interested in the science, which is fine, but are we going out enough?
Yeah, so I ask myself that a lot too, because anytime I write anything, right, I'm like, okay, this is like a point I really want to make, but I'm preaching to the choir. Like everybody else believes that too, for the most part, that follows me. And so that reach isn't necessarily as broad as you would want. So I think that is a challenge, especially on Twitter, because you can kind of pick and choose, right, who follows you. I think in some ways other social media platforms are a little bit more broad, like Facebook, because most people have also like family members where they don't all agree on stuff.
Yeah.
So sometimes that at least like breaks out of that kind of ideological bubble. I think that Twitter tends to be a very much, just different ideological kind of bubbles that just echo each other.
Yeah.
And I think the best way to kind of try and break out of that is when you're tweeting about something, right, try and make it accessible, like especially when it comes to people not wanting to trust scientists, or being like, oh, you have a PhD or you're a scientist and you think you're, way up here on this ivory tower, and like you don't care about, me and people like me, and trying to like really kind of humanize science.
Yeah.
Because I think that helps, like, oh, that scientist is like me, like she believes some of the same things that I do. Maybe we don't agree on everything, but like I get where she's coming from on this, and really trying to be broad in that sense, because they'll still pick it up on their feeds, right, if other people that they follow like it. And if you can humanize yourself, right, and not just be like, this is an important finding and blah blah blah, and this is why this is amazing and whatever, my parents and my family wouldn't like look at that and be like, oh yeah right? Like, okay, but why does it matter to me, right? And which makes total sense. And I think we need to connect the things that we're trying to send out to people. We need to make it more personalized almost to them. So
I agree. There's a big movement in science is starting to do science comedy, and I'm a big supporter of that. I introduced that in an association I volunteer with, And the whole idea behind that is exactly that. You want to humanize scientists. We're not up there in the ivory tower. We have funny incident at the workplace. So we suffer from the same things people are suffering every day. The fact that we have some sort of knowledge of whatever that matter, if anything, makes these things more funny, because you can really see that despite you can do whatever you want, but you always encounter these sort of difficulties in life. So I think it's I completely agree with that point. Well, as I said, I don't want to steal too much of your time, and this was already fantastic. If I keep on thinking about what you're doing, I can already figure at least another 10 questions that I could be able to ask you. So I guess we'll stop it here. Can you tell us where people can reach? I guess Twitter, but feel free to shout out anything that could...
Yeah, you can reach me. Probably the most stable right now is Twitter. So my handle is OAmbiguity, OHAMBIGUITY. I had a lot of feelings in college, I guess, when I made that. But also I'm interested in anxiety and ambiguous stimuli. And so
which makes sense.
It fits very well, I think. But yeah, you can find me on Twitter. You can also reach me on email, or my website has a contact as well, which is jenniferhoneycuttphd.com.
Okay, Jennifer, thank you very much for this time. Congrats again for expecting the baby, and good luck with that. I'm sure it'll do great.
Yeah, thank you so much. I think this is a great opportunity. I'm really excited to see what you get done with it.
Yeah.
Thank you for listening to the interview, and I hope you enjoyed this episode of Draw My Science. And if you did and you would like to support the show, you can of course like this video, subscribe to my channel, or visit my shop, link in the description, where you can find products with the illustration I make. I hope to see you again in the next episode. Bye!
It's a nice way to break up the day.
For me, I'm starting the day now, so I still have to get up, but it was a good way to start my day.